Good morning, good afternoon, and good evening, everyone. My name is Jocelyn Basile, and I'd like to welcome you on behalf of the University of Sydney Infectious Diseases Institute, or Sydney ID, together with the Australasian Society of Infectious Diseases, the Australian Veterinarians and Public Health, and Queensland Health. I'd like to start by acknowledging the traditional custodianship of the various lands on which we meet today, and to pay our respects to those who have cared and continue to care for country. Today, we have 4 absolutely brilliant experts joining us, and I'll leave our organising committee to introduce each expert as they speak. I wanted to acknowledge and to really thank our organising committee, Ruth, Kate, Michael, Catherine, and Rebecca, for all their efforts in putting together this webinar. Please use the Q&A box for questions. Please contact us anytime via email. If you'd like to join the conversation on Twitter, please tweet using the hashtag QFever, and please tag our organisations as well. And on that note, to begin, I'd like to pass on to Kate to introduce our first speaker. Over to you, Kate.
Thank you, Jocelyn. Well, it's my very great pleasure to introduce Dr. Ben Bauer to you tonight. Ben studied veterinary medicine at the Ludwig Maximilian University in Munich. During this time, he also embarked on a study program abroad at Massey University in New Zealand and obtained a postgraduate diploma of veterinary clinical science. After his return home, he completed his veterinary degree in Munich and received his Dr. Med Vet. He has passed his exam to become a diplomat of the European College of Small Ruminant Health Management, which is a European board veterinary specialisation. Currently, he works in the Clinic for Swine and Small Ruminants at the University of Veterinary Medicine Hanover Foundation, and his research focus is on control of Coxiella burnetii in small ruminants in the spirit of the One Health approach. And we're very excited to welcome Ben to Australia later on in the year. This evening, he'll present on Q fever outbreak on a dairy, goat and cattle farm, an example of the One Health approach. Thanks, Ben.
Yeah, thank you, Kate, for your kind introduction. And I just want to share my screen now. So I just give me a short feedback if everyone can see my screen.
Yes, Ben.
Yeah, thank you so much. Yeah, thanks again for the kind introduction and the invitation. So my topic today is a Q fever outbreak on a dairy, goat and cattle farm, an example of the One Health approach. So that's a case report I would like to present to you. And before we jump into that case, I just want to give you short recap of Coxiella burnetii and Q fever. So it's an intracellular bacterium with antigenetic phase variations. The main reservoir for Coxiella burnetii are ruminants, namely sheep, goats, and cattle, and infected animals shed the pathogen mainly during abortion and parturition through birth products, but also through milk and feces. Coxiella burnetii is highly resistant against desiccation, UV light, and many disinfectants. So the pathogen is detectable in dust and wool for up to 2 years, and dry and windy weather conditions favor the spreading. It is mainly worldwide accepted that Q fever is an airborne disease, but the oral route of transmission and also the transmission by ticks is still under discussion. It is a zoonotic disease. It's highly contagious, so the ID50 is only 1.18 bacteria. So I take you to a farm we took care for almost 5 years. And this farm is located in northern Germany. Just to give you a short overview, here the UK, Denmark, and Germany. And I'm located here in Hanover, and the farm is close to the Danish border on that peninsula. The special location plays a role later in our story. So this is the farm. It's a dairy cattle and dairy goat farm. And this slide just symbolizes the location of the barns. So there are around 360 dairy goats on that farm and 95 dairy cattle. And the barn are really, really close. They are just separated by a wooden wall. And also the doors are very close. So the farmer called us for more than 5 years ago because he had some problems with his dairy goat herd. So several abortions and weak kids were born. And at necropsy, Coxiella bonnetii was identified as the only abortifacient agent on that farm. So a CQ value of 9 means that's a really, really high amount of Coxiella bonnetii around. So we went to that farm and we collected vaginal swabs from 63 goats, and they were all tested positive with a high amount of Coxiella bonnetii. From that samples, we were able to do some genotyping. So we genotyped the Coxiella bonnetii isolates with the MILVA method. That's a special genotyping method. And we could identify a cattle-associated genotype called C16. That was quite new. So that was a new genotype we identified on that farm. Moreover, we performed phase-specific serology with the goats, and we could identify dominance of IgG phase 2 antibodies, which reflects an acute infection in that dairy goat herd. So we did also some tests with the dairy cows. We also collected vaginal swabs from the dairy cows, 22 in total, and all vaginal swabs were also tested positive. And we performed also genotyping, and we found the same genotype as in the dairy goat herd, in the dairy cattle herd, so again, C16. We performed again phase-specific Coxiella bonnetii serology, and we found same levels of IgG phase 2 and phase 1 antibodies, which reflects an ongoing or already a past infection. Interestingly, during our farm visits, we saw the farm cats and the dog eating placentas, and they drink raw milk from that goats. So we decided, oh, that's quite interesting. We should really take the cats and the dogs also in our investigations. So we sampled also the cats and the dog on that farm, and the 3 queens also tested positive by the vaginal swabs, and 4 out of 9 cats tested seropositive. And also the dog was seropositive, but the vaginal swab tested negative. So after Q fever was diagnosed on that farm, all ruminants were vaccinated with a Coxiella bonnetii phase 1 vaccine, which is licensed in Europe and several European countries. But we were also interested not into the animals, but also to see what's about the family. So the farmer's family consisted of father, mother, and 3 children. So colleagues from the medical department did the blood sampling and analyzed the blood samples with phase-specific serology, namely indirect fluorescent antibody tests. And particularly the mother showed an antibody response, mainly an IgG phase 2 response, which is interpreted as a recent acquired Coxiella bonnetii infection. And that fits actually to her medical history, because in January 2018, when the Q fever outbreak was detected in the dairy goat herd, she suffered from flu-like symptoms and recovered after the treatment of doxycycline. So that's also an indication that, yeah, that was probably Q fever. And she also performed high-risk activities on that farm. So she was responsible for the daily milking and the obstetrics on that farm. So I really like that slide because it's summarizing all our findings and putting them in the epidemiological context. So we assume that Coxiella burnetii was shed by the dairy cows, and they infected the pregnant dairy goats with the genotype C16, the new genotype, and that led to a huge release of Coxiella burnetii during kitting season in the dairy goat herd, and led to abortion. And so at least one family member got infected. And also the cats and the dog got probably infected by the dairy goats. And what we could not focus on is the role of the cats, because in the past there were some Coxiella burnetii outbreaks associated with cats. So it seems to be that cats have a special epidemiological role in spreading Coxiella burnetii, for example, to neighboring farms, but we were not able to follow up this. So as I mentioned at the beginning, we have this special location of that farm, and on the peninsula in the North Sea. It's a bird sanctuary with dikes and marshland. And what makes that area very special is that we have a high density of sheep. So almost 109 sheep per square kilometers. So that's 5 times more than the average state level. In that area, we have a traditional pasture-based production with 2/3 agricultural land. And so because of this high sheep density, we were quite afraid that we are now facing another Q fever epidemic like in the Netherlands. Maybe you know that, are you aware about that in the Netherlands, so Coxiella burnetii spread from one dairy goat farm to the next. And we just thought, okay, will be that also happen now in that area, on that peninsula, spreading Coxiella burnetii from that dairy goat farm to the sheep in the surroundings. So therefore, we asked farmers to participate in a special, additional study within a 10-kilometer risk area. And 12 sheep farmers agreed to participate at that study. So that represents around 6.6% of the sheep farms within the risk area. Here again, you see on the right side, the peninsula, the star symbolized the positive goat and cattle farm, and A2L are the sheep farms. And the square on the left side is the weather station, which plays a critical role later. So we went to the sheep farms in spring 2019, and we took samples, serum samples and vaginal swabs from ewes which lambed within 8 days. And to examine the environmental contamination, we decided to take dust swabs from the sheep farms. So we used the swabs rolled over 1 meter window seal, and also checked these dust swabs for the presence of Coxiella burnetii. And we were really quite surprised, because all sheep tested, there are more than 350, were tested negative by PCR with the vaginal swab. So none of the sheep shed the pathogen. There was only one positive dust swab from the sheep barn. It was on farm K, so more than 8 kilometers away. And we could only identify 2 farms which are seropositive, so B and D, with a really low amount of seropositive sheep. And we found it really astonishing that farm A, which is only 80 meters away from the Coxiella burnetii positive dairy farm, was totally negative. So to go to that positive dairy goat farm, you have to pass farm A, and there's lots of exchange going on. But it seems to be Coxiella burnetii was not spread to that farm. So we were really wondering why that was the case. And we decided to take a closer look on the meteorological situation during the time, during kidding season, and therefore we analyze data from the local weather station, which are freely accessible at the internet, and we consider the wind direction, the wind velocity, the precipitation, and the relative humidity from January 2018, when the Q fever outbreak occurred, until April 2019, end of our study. And these are the wind direction and velocity during the kidding season. On the left side, we have this compass rose demonstrating the wind direction and also the wind velocity. And the colors here, the brighter the colors get, the stronger the wind gets. And during kidding season from January to April 2018, around 30% of the wind came from eastern direction, and more than 50% of the wind velocity was quite strong. So we had more than 5.5 meter per second, which represents a moderate breeze until storm. And you can compare that with the location of the farms. So 30% came from eastern direction and probably drifted Coxiella bonnetti into the North Sea. And this is just summarizing the analysis from the time period May 2018 until April 2019. So main wind came from western parts, but again, a really high wind velocity. And these are the results from the precipitation and monthly relative humidity. So on the y-axis, on the left side, we have the precipitation in millimeters, and the bars symbolize each month. So we have from January 2018 to April 2019, we have the kidding seasons, and we have here just to have a comparison if there's a high precipitation or low precipitation, and that line, the 10 years average of precipitation. And during the kidding season 2018, there were at least high rainfalls in January and April, and then afterwards, the peninsula really suffered from a severe drought. So it was really, really dry, but this ends again in kidding season, during kidding season 2019. So at the beginning, we had a really high precipitation with the exception of February 2018. And we had also high humidity. So on the right side, we have the humidity, and the dotted line represents the monthly humidity. And again, so it was over 80% during the kidding season, and then dropped down under 80% and rose again in October. So taking all this into account, this is just some possible explanations for that. So this is just some basic work, what we have done regarding the weather situation there. But what we can say is that during kidding season 2018, during that Q fever outbreak, so easterly winds probably drift Coxiella bonnetti in the North Sea, so it's gone. So we had also probably due to the high precipitation and high rainfalls, a high humidity, and so that prevents probably the formation of dust and aerosol dispersal. So that prevent probably the spreading of Coxiella bonnetti. Also grassland and dryness stitches that probably prevent the formation of Coxiella bonnetti contaminated dust. There's a hypothesis from colleagues from the Netherlands saying that high wind speeds will have a dilution effect on that. That could also be the reason why Coxiella bonnetti was not spread to the sheep farms. And despite we have that favorable conditions during the summer months, because we had really this drought in summer 2018, would be easy to transmit Coxiella burnetii from the dairy goat farm to the sheep. But it obviously did not really happen. So because no pregnant sheep were around at that time, because Coxiella burnetii seems only be successfully infect animals if they are pregnant. So I'm coming to our main conclusions of the whole case. The phase-specific zeology and milva genotyping are helpful diagnostic tools to characterize Coxiella burnetii infections in ruminant herds and to identify the potential source of the pathogen. We consider cattle herds as potential reservoirs capable of spreading Coxiella burnetii to small ruminants. So this is particularly quite important under German conditions, because many German dairy cattle farmers blame shepherds for spreading Q fever, which is obviously not the case. So dust swabs are a quick and inexpensive diagnostic tool for testing ruminant herds for the presence of Coxiella burnetii. So in that case, we were able, or we were not able to identify Coxiella burnetii on the farm. At the moment, we are just focusing on that approach, using dust swabs for surveillance of Coxiella burnetii in ruminant farms. And last but not least, inclusion of meteorological data in risk assessment for Q fever outbreak is necessary. This is also quite important to fulfill the demands of One Health, which is taking animal health, human health, and the environment into account. And if you are interested in more information, just visit our website, www.q-gaps.de. So we are collecting all our data of our Q fever projects on that farm. We are also providing some flyers for the general public and also for human medicine to inform people and colleagues about Q fever. And at the end, I thank you for your attention.
Wonderful. Thank you so much, Dr. Ben Bauer, for that great presentation of a really nice epidemiological study, including the environment as well as our human and animal counterparts. Now, I know there's a lot of questions in the chat room, but Ben will be available to answer those later on at the end of the session. So it seems that maybe the answer is blowing in the wind. I'm sorry, somebody had to say it. Now, our next speakers are Doctors Candice Holland and Robert Horvath. Candice is an infectious disease physician working for Westmorton Health in southeast Queensland. This is servicing a semi-rural area west of Brisbane with a high Q fever caseload, so she's got her work cut out for her. Rob is an infectious disease physician at the Prince Charles Hospital in Brisbane, and also a microbiologist for Pathology Queensland. His career has spanned Queensland, New South Wales and the ACT. He's co-founder and co-chair of IEQ, which is Infective Endocarditis Queensland. He's also very busy as the co-founder of ACE, which is the Australasian collaboration in endocarditis, and also, importantly, co-founder of QFIG, which is the National Q Fever Interest Group. Together, they're going to present today on the activities of QFIG, as well as give us some ideas about the epidemiology across Australia of human coxiellosis. So I will hand over to our speakers. Thank you.
Oops. Oh, hello. Oh, I don't know whether, it's the camera not working. Just a minute. Whoops. Yeah, sorry. Anyway, now after that amazing scientific talk, a slight change in tone, I guess, but whoops. This, I think you can see my talk.
Not yet, Rob. If you're able
Okay,
to,
why is it not doing it? Oh, okay.
we can, excellent. And if
Can you
you
see
just,
it?
yes.
Oh, you can see it. Oh, good.
Um,
Okay. Right. So
thank you.
anyway, thank you everyone for giving us the opportunity to enlighten you about QFIG, an innovation that potentially may change our entire knowledge of Q fever, with local data to improve outcomes significantly by the centenary of this disease's discovery. You know, to have the opportunity to give this talk so recently after QFIG's birth is truly humbling. Firstly, back to where it all began 90 years ago. Cannon Hill Abattoir in Brisbane, which caused a tsunami of illness in its workers due to its practice of slaughtering pregnant cows. A tsunami that necessitated a large-scale investigation that resulted not only in the discovery of the organism, but also the introduction of nomenclature used to this day. Coxiellois burnetii after its co-discovery, Sir McFarlane Burnett, the person on the left, and Q fever, initially Queensland fever, a name vetoed by the Queensland government as it was judged bad for tourism, and changed to query fever as a compromise to keep the letter Q. And being asked to give an origins talk, I thought about the Marvel franchise, where every character has an origin story. This character symbolises QFIG with its origin story. However, if you want to hear about my role in the origin story, this is a more accurate depiction. A very implausible but true chain of events is about to follow. Strap yourselves in. The very beginnings of QFIG were in 1996, where as a registrar in Newcastle, I not only diagnosed my first ever case of Q fever endocarditis, but also did a vaccine clinic in the abattoirs of the Hunter Valley, where my role really was to authorise vaccinating anyone the Q fever nurse wanted to vaccinate. Was not only a valuable lesson in the intricacies of skin tests and serology to judge prior infection and the likelihood of vaccine misadventure, but a surprising number of people turned up to those clinics who didn't work in the abattoir but were willing to pay themselves for the vaccine. However, interestingly, most of these people already had antibodies, a hint that this infection was endemic in parts of rural Australia. After this birth by fire into the Q fever world, the next significant year in the naissance of QFIG was 2006, in the least likely place on the planet for a Q fever milestone to occur, the only time you'll ever see a photo of New Zealand during a Q fever talk. If you think the location is weird, the next 60 seconds are only going to get weirder. 2006 was the year ASID invited Didier Raoul to speak about Q fever at the Wellington ASM. I was working in Prince Charles by this time and had been treating several cases of Q fever endocarditis with results quite different from Monsieur Raoul's papers, and was keen to hear from the guru what I was doing wrong. However, Monsieur Raoul inexplicably only briefly appeared, refused to talk about Q fever at all despite being on the program, and evaded Jenny Robson and myself's attempts to ask his opinion on what we were doing wrong. After that Weirderama episode, I began to think the problem was that it was perhaps a different disease in Europe to Australia. As my senior colleague at the time had a research interest in Q fever, I didn't want to tread on his toes and pursued other interests. Jump another decade to 2015. I was now acting director at TPCH, so the door was now open to follow my passion of Q fever endocarditis. TPCH already had the largest published series of Q fever endocarditis in Australia, but that trial was decades old, before echocardiograms and when rheumatic heart disease was still very common. Luckily, our reg at the time, Mark Armstrong, was keen to assist, and we collated the largest modern series of Q fever endocarditis in Australia, which did interestingly demonstrate some discrepancies from the French data. My first thought after this was, we need more Queensland cases with a statewide study, but the extra numbers were judged not enough to justify the effort, so I parked the idea. Still playing around with the idea of enlarging the study set, the next milestone was in 2018, when I had the opportunity to have a long chat with Stephen Graves at a microbiology conference in Q fever's birthplace. Stephen mentioned his passion for a new vaccine, and I said I'd see what I could do to help, and the first interstate Q fever partnership commenced. Then things accelerated. 2019 was perhaps the most pivotal year for QFIG, as a chance meeting with Sarah Coghill at Darwin ASID had us both chatting about our Q fever experiences. and the idea to have a multi-state, multi-centre collaboration on Q fever, both for management of complex cases and for research to establish what is best for Australian patients began. This was a truly serendipitous moment, as my mindset that ASID was actually just to promote an endocarditis symposium I was organising, which coincidentally led to the formation of ACE. So arguably, Darwin is the birthplace of both ACE and QFIG. Fast forward past the early COVID years to 2021, when the deferred zoonoses meeting was held COVID safe as a webinar. I spoke about Q fever with an emphasis on endocarditis and lamented the need for an interest group to answer our burning Q fever questions. Positive emails and a feedback from that session led us to advertise a Q fever interest group national telemeeting. 33 people of various occupations from across Australia participated in an amazing brainstorming session. I believe Kate Garnham came up with the QFIG acronym. Candice voiced her enthusiasm with extensive public health contacts, and the rest of the steering committee was formed. The rest, as you may say, is history. I was absolutely staggered by the response and enthusiasm and the sharing of a vision to meaningfully improve our understanding of Q fever in Australia. Although October 21st, 2021 is the official birthday of QFIG, it was conceived earlier and the product of a very implausible gestation. It currently has 70 members representing in alphabetical order like the Olympics, academic researchers, anatomical pathologists, cardiologists, GPs, infectious diseases physicians, microbiologists, public health physicians, vaccinologists, and vets. And the list is just getting longer. Every mainland state is represented in our membership. Our aspiration is that by the time Q fever celebrates the centenary of its discovery, and note airborne transmission, Q fever will have changed our understanding and management of Q fever unrecognisably to the benefit of the estimated up to 2 million Australians exposed to Q fever, with data that may improve Q fever management across the globe. And before I hand over to the real person who keeps QFIG going, Candice, I'd like to thank firstly the steering committee, Sarah, Kate, Stephen, Penny, Jenny, and Nick. And I'd like to thank organisations like ZUSIG that have faith in our vision. And mostly I'd like to thank our members, without whom none of this vision would be possible. And the door is still open to anyone who'd like to join, perhaps the largest collaboration in Q fever research and management Australia will perhaps ever see. Thank you, and hand over to Candice.
Thanks, Rob. Just briefly going over the epidemiology in Australia for human cases. So Q fever is notifiable in Australia throughout the various jurisdictions and under their Public Health Act. So this is a graph looking at notifications across the period of 1991 up to 2022. This is collected by the National Notifiable Disease Surveillance Service. And you can do these interactive dashboards with the data, which is very handy to look at this in different ways. But just to point out from this slide, it's a male predominance, which is likely to be reflective of the industries and exposures of people at risk of Q fever. And you can see very much that it's in that working age range. So people who are occupationally exposed are the predominant risk group. Just to the next slide, please, Robert. And then just breaking that down by jurisdiction in the, again, this is the same time period, 1991 to 2022, but longitudinally this time. You can see that New South Wales and Queensland in the purple and blue lines have by far the bulk of the notifications, so around 48 and 39% of notifications respectively. And the other thing that you can appreciate from this graph is a significant drop in notifications from 2001 to 2006, and this coincides with the Australian government's national Q fever management program, where Q fever vaccinations were subsidised for people who were at risk due to their occupational exposure. So on standing down of that program in 2006, there has been a steady increase in the incidence. So this database, I should mention, captures acute Q fever notifications. Just to the next slide, please, Robert. And then just to present that geographically for those that are not aware of the geography. So the distribution really fits with the animal husbandry and farming practices across those regions. So central, west and southwest Queensland and northwestern New South Wales. Just to the next slide, please, Robert. So in terms of the QFIG activities to date, we were a little bit slow to get going because of COVID and other time commitments of the various members, and trying to actually coordinate the diversity of people that Robert has conveyed to you. It can be very difficult to align schedules, but we based on that initial brainstorming exercise, we distilled down the ideas into 5 particular discrete pieces of work to begin with, at least for the group to get going. This was done by sending out expressions of interest to the whole membership group, and then a selection process by the core membership to determine the best mix of people with their skill mix, and also trying to get that geographic representation across the different jurisdictions to make a really informed look at each of these pieces. So there were 4 working groups and then a major steering committee. So the first is to look at aligning our surveillance for the public health notifications. So trying to ensure that all of the jurisdictions are collecting the same data essentially for the acute Q fever cases. The second is to look at doing our own Australian Q fever clinical practice guideline. This is likely to evolve as the information comes through from the other pieces of work, and you'll see that these things all feed into each other. A laboratory study, which is headed by Jenny Robson with Sullivan Nicolaides Pathology and Stephen Graves with the rickettsial reference laboratory, and cross-section of clinical microbiologists across the different jurisdictions, and then a retrospective data linkage study to see what data sets do we have at the present time that we could use to answer some of the questions, at least, while we look to the big piece of work, which is on the next slide, please, Robert. This is, I guess, the pride and joy of QFIG and our major project, which is the establishment of a national clinical Q fever registry. So this is looking at human clinical cases at this point in time, but it would be good to move into a more One Health surveillance database as time goes by, and that's the aspiration. But it's a little time away yet before we'll get to that level of data. Just to the next slide, please, Robert. So just some other miscellaneous activities that we're doing through QFIG. So some advocacy work. At the moment, the Q fever vaccines are held in a registry by industry, and I think that's one group that we should look at in terms of expanding for QFIG membership, is to have a little bit more industry representation and workplace health and safety. But we are drafting a letter to advocate for inclusion of the Q fever vaccines onto our Australian immunisation register as a mandatory vaccine listing, because it is quite difficult to get that information when it's held in sort of an industry database. To the next slide, please, Robert. And then finally, the value of having such a good network is to be able to gather together the group of experts and discuss through challenging clinical cases where there may not be an answer in the literature. There's still a lot of query in Q fever, and so at least we can bring together people who have varying levels of clinical experience and exposures to be able to talk things through and get the best outcomes for our patients. Next slide, please, Robert. So we've got, there's many possibilities for the future. We really wanted to be very focused and make sure that we do progress some of these activities in the first instance. So it's really important to be quite defined and consider what the hypotheses are that we're trying to test with each of these activities, and ensure that they're linking in with each other. But things that we'd like to explore into the future include that more One Health focus on the surveillance, being able to do, like the first presenter had showed, a more One Health informed risk prediction to be able to know which groups to target in terms of prevention activities, to progress our knowledge on vaccination and explore new vaccinations, but also, again, across both human and animal sectors, and to look at Q fever in pregnancy, in particular in humans. Robert, I'll let you jump in here with your point about testing.
Yes, one of the things that QFIG has enabled, which we didn't think possible before, is what we call the enhanced testing. And the other thing that's fallen into play was being able to use the PCR analyzers that normally would have been used for COVID to have a very rapid turnaround time. Sometimes I think the record they've sent to me is just over 2 hours between receipt of the sample in the laboratory and a result, which is absolutely amazing for Q fever. Because until about 2 or 3 months ago, you're waiting a week for a PCR on average, and you are in most jurisdictions in Australia. But since they've rolled out as a temporary, but it looks like it may be permanent from the discussions going around, hopefully rapid turnaround test, even though the numbers are still small, we're finding it's over 10% and approaching 15% positivity, which is an amazingly high positive rate from what we call, testing people that we wouldn't normally have even bothered requesting a PCR or sometimes Q fever testing on. So I think we're discovering a lot of that we're way underdiagnosing Q fever. So that we may have a bit to see in the next few months from this. It may change our entire standard of care with how we look for Q fever in humans, and how we test for it as well, who we test for and how. So with that improved knowledge, with being more accurately able to test people, particularly very early in the course of the illness now, instead of a week or two down the track, may give us information about how we can actually alter the outcomes and potentially reduce people going on to chronic Q fever with any luck. But yeah, it's an interesting new phase that we're going from. This is just one of the multiple things, as Candice has outlined. We haven't discussed all of them, but these are the ones we're moving with the most right now.
Yes.
So back to you, Candice.
Yeah, so I think our members have a lot of questions, and same internationally, about natural history. What are the geographical differences? Australia versus other countries, but even different areas within Australia. The predictors to go on to chronic Q fever. Who should we screen? Do we have biomarkers to know who's going on to have chronic Q fever and other complications? How do the investigations progress over time? And to answer questions about the Q fever fatigue syndrome and response to interventions. So these are some of the questions that we have and that we're hoping to answer from these various pieces of work. To the next slide. Thanks, Robert. So just put up our website address and the email address for any persons who would like to be in touch, or if you have any questions for Robert or I or other members of the steering committee. Thanks.
Thank you very much, Candice and Robert, for a very interesting and very different perspective on Q fever. There are some questions in the Q&A already that Rebecca will moderate later. Before we get to Q&A though, we have one more presentation, and that takes us to a different part of the world in a different setting yet again. Our final expert is Dr. Jo Halliday, who is a senior lecturer in zoonosis epidemiology at the University of Glasgow, and does a lot of her work in Tanzania and takes the most beautiful photos. So it will be a feast for the eyes as well as for the brain, I think, this presentation. Jo has over 15 years of experience undertaking field investigations of the epidemiology of zoonotic pathogens at the interfaces between humans and domestic or paradomestic animal populations, and she'll show images of what paradomestic animals look like. Her work is actually focused on a set of endemic bacterial zoonoses, mostly those causing acute febrile illness in people, so slightly different presentation than the chronic one we heard about in the previous talk. And those endemic bacterial zoonoses include not just Coxiella, but also Brucella and Leptospira. So if anybody's planning another webinar on zoonotic bacteria, Joe's your person. These bacteria cause human illness and livestock productivity impacts globally. And there are still fundamental gaps in our knowledge regarding transmission, the impacts of infection in different populations, and the potential impact of control strategies. And to start picking away at those knowledge gaps, Joe will tell us about Coxiella as a multi-host pathogen in Tanzania. Thank you very much, Joe.
Thank you very much, Ruth, and to the organisers, and thanks for the invitation to talk today. Can I just check that you can see my slides and hear me okay?
Yes, I can, Joe. Thanks.
Great. Okay, so yes, so thanks again. I'm based at the University of Glasgow. I'm going to give a bit of an overview of work of many people in many places over a series of projects in northern Tanzania. So I'm just going to talk a little bit about the One Health surveillance platforms we've set up, a little bit on livestock prevalence and risk factors, and then disease impacts in terms of an abortion surveillance network. Also bring in some human prevalence and risk factor data, and a little bit at the end on where we are and are heading on molecular data and comparisons across populations. So these two photos represent what I'm going to call the pastoralist context in northern Tanzania, and I'll talk a little bit about the different systems. But just as a sort of overview of where a lot of the work of this group started in terms of the understanding of Q fever in Africa. This graphic comes from a systematic review published in 2014 from the wider project team on a study called Bacterial Zoonoses, and illustrates a pretty small number of studies from Africa that made it through the screening processes, but that demonstrate serious surveillance studies in animals and people, but also disease in animals and people, and an evidence of Q in all of those populations. If you can see this square here, this is a study in a febrile population that I'll talk more about in northern Tanzania, led by John Crump, in which about 5% of individuals presenting with acute febrile illness were diagnosed with acute Q fever. And that's through diagnostics that are not routinely available, and most of that clinical burden is routinely invisible. But the sort of overall story from this review was evidence of endemicity of Q fever or Coxiella in cattle and small ruminants across the continent, but seroprevalence showing quite highly variable patterns by species and location, little understanding of what drives that variation or the risk factors, and that we knew very little about human incidence. And we also knew very little about the sort of production impacts of Q fever in any of these livestock populations. So my involvement in all of this started with the Bacterial Zoonoses study, which ran from 2011 to 2015. This is a photo of the launch. I won't be able to mention everyone who's contributed throughout, but I'll just highlight Sarah Cleveland, who I think might be listening in, John Crump, Catherine Allen, Ludovic Caswala, Ben Marrow, Blandina Mbaga, who's not on this, and many others who have been involved in all of the studies that I'm going to give an overview from. And this study started with the goal of determining prevalence of Coxiella burnettii among linked animal and human populations across different agroecological contexts, then looking at risk factors and determinants of exposure and disease risk in those populations and how they varied by system, and trying to get a handle on understanding burdens, ultimately to try and inform future control strategies. And those overarching goals have applied in the second and third generation projects as well. So just to talk about where we are, so this is Tanzania within Image Africa, and then I'm talking mostly about data that comes from the very north of Tanzania. So this is the Kenyan border. This red marker indicates Moshi, which is the location of the Kilimanjaro Clinical Research Institute, sort of the host in Tanzania for much of the work that I'm going to present on. And then this star just shows the location of a clinical hospital called Engelin that's within the area of the Ngorongoro Conservation Area, where we set up some additional febrile surveillance that I'll come back to. Zooming in a little bit, so this is that northern Tanzanian context again. This is the Kenyan border running across the top. And this just visualizes all of the different platforms and data resources that I'm drawing from to give this overview. So this shows the outlines of regions of Arusha, Kilimanjaro and Manyara in northern Tanzania. All of the little areas within that are wards, which are administrative regions. And we've got 2 sites that I'm going to tell you about human febrile surveillance from. Most of that work's been done, as I say, at KCMC, which is in Moshi, and there's several generations of febrile surveillance work led by John Crump, Matt Rubach and teams there. This is the location of the Enderlin Hospital on this map, where we ran a febrile surveillance platform in a predominantly in a hospital serving a predominantly pastoralist human population. The red dots on this map show the sites of sampling of cross-sectional studies of livestock and people through 2 studies called BACZOO and SEEDS. So those are household-based samples. And then the yellow shows you where the SEBI study generated data on abortion etiologies. And so those are a subset of the wards where we had undertaken cross-sectional sampling. So we've got these overlaying layers of information from multiple studies telling us about livestock and people and their interactions. And I'll tell you a little bit about each of those things. So starting with the livestock and the serious surveillance that was done in 2013 to 2014. This is the BACZOO cross-sectional study that I mentioned. So it's a stratified random sample, selecting a number of wards that represent these settings that we defined a priori as pastoral. So predominantly Maasai pastoralists managing extensive multi-species livestock herds, so cattle, sheep and goats managed together and extensively. Agripastoral, which is sort of an interface area where communities are using livestock, but also crop production for income generation and subsistence. And then a peri-urban population, which is livestock, smaller numbers of livestock kept on the edges of urban areas broadly. The classification of these systems is something we go into in great detail, but I'm not going to in this talk, but I can point you to the right place. So we had these 6 wards in all of these contexts, up to 15 animals of each cattle species, cattle, sheep and goats sampled in each of the households that were randomly sampled within that. We also sampled people who were willing to give a blood sample in those households. All of the livestock serum was tested by ELISA with the LSI Vet ruminant kit, and that was done at Morden or Glasgow. And then when there were more than 15 animals per household, we preferentially sampled adult females, and vaginal swabs were collected, total DNA extraction, and then the IS 1111 target for qPCR, which Ben's already mentioned, was run on those swabs. So this gives you an overview of all of that data, and I'll walk you through it. So on the y-axis, we've got the ELISA titer score, and the dotted line shows the kit cutoff. So points above the line are considered positive for Q fever in these livestock species, and below the line negative. Within each panel, we've got BO are cattle, CP goats, and OV for sheep, and the points are jittered just so that you can see them within those categories. The panels show the peri-urban context here on the left, agripastoral population in the middle, and the pastoral population on the right. And then the final thing is that the point color shows you, as well as the ELISA score, the PCR result from that same animal. So individuals that are represented by green dots are PCR negative, and the orange dots are PCR positive individuals. So you can see that we do have exposure, particularly in goats, across these different systems, but there is much less of it in the peri-urban and agripastoral populations, and essentially very little, if any, PCR positivity in those systems. Whereas it's a totally different story. There's much more exposure and there's much more PCR detection in the pastoral systems. So putting that all together as overall prevalences, this is quite a busy slide, but all in, across all those different systems, we've got about 8% seropositivity in cattle, coming on for 20% in goats, and about 17% in the sheep. And the modelling of these data, unsurprisingly, goats and sheep are much more likely to be positive as compared to cattle in the pastoral system versus non-pastoral systems, where we're much more likely to find seropositives. The equivalent data for the qPCR, we've got about 4% of cattle, and these are routinely sampled cross-sectional animals. There's no clinical event associated with this sampling. So about 4% of animals at home have detectable DNA in their vaginal swabs. That's about 10% in goats and getting on for 15% in sheep. so consistent with other areas. And again, goats and sheep versus cattle, much more likely to be PCR positive. More PCR positivity in the pastoral versus the non-pastoral areas. And as you can see, going back a slide, we've got PCR positives in ELISA positive individuals, but also lots in ELISA negative individuals. The ELISA score is a good predictor, a significant predictor of PCR positivity. So animals with higher ELISA titers are more likely to be swab positive. But if you look at the relationship between those two tests as positive negative classifiers, the qPCR and the ELISA, the agreement is slight. So we've got lots of shedding by seronegative animals, and we've got ELISA positive animals that are not shedding, which is consistent with the sort of intermittent pattern of DNA detection seen in other contexts as well. So in terms of the risk factors overall, this is work driven by Will de Glanville, and he's used data that come from all of the cross-sectional populations. So these two different studies, it's called SEEDS and BACZOO. And then this graphic just sort of visualizes that gradient of risk across this sort of scale from pastoral to peri-urban. And on the y-axis here, we've got the marginal predicted probability of seropositivity in a ruminant. So this is spanning cattle, sheep, and goats. And then we've got this gradient that he's represented with a continuous scale in this analysis from pastoral to the peri-urban or smallholder end of the spectrum. And we've got a declining probability of seropositivity in those livestock species over that gradient. So the setting is really important. Risk factor analyses is ongoing, and we're working on sort of disentangling lots of correlated variables in these analyses. But we've got increasing age of the animal, unsurprisingly, contributing to greater chance of seropositivity, and then also a series of environmental variables. So increasing temperature and increasing rainfall are associated with seropositivity, decreasing tree cover, but also decreasing wind. So some of those variables that Ben mentioned are associated with the probability of exposure in these populations, but not all in the directions that we'd expect. And we're working on those analyses, as I say, and I think some of these relationships, teasing them apart, complicated by the quite long persistence of antibody positivity and changes in these climatic variables over time, and that complicating seeing how we interpret evidence that's based on exposure rather than recent cases. But we are seeing definitely that these environmental variables are influencing the animal exposure pattern. Coming on to sort of impacts in terms of production impacts in livestock. So these data come from a study called SEBI, which ran in 2017 to 2019. And these are the yellow polygon areas in the earlier map. So this was a prospective cohort study of abortion event reporting with reactive sampling and diagnostic follow-up. And it captured 215 abortion events. And from those, vaginal swab samples were collected from the dam, exterior fetal swabs, and placental tissues as well. All of which were tested by the IS 1111 coxiellois PCR. And the samples analysed came from 99 goat abortions, 45 sheep abortions, and 70 cattle abortions. And this work was all largely driven by Catherine Allen, Kate Thomas, and others. And these are the findings that they had. So in terms of detection of C. burnettii in materials from those events, about a quarter of the animals, a quarter of the events, one or more samples from each event was positive for C. burnettii detection. But we're bearing in mind, of course, that we've got non-aborting dams in the cross-sectional population that show a lower, but still detectable proportion that are the positive in the same assay. So what the study tried to do was sort of attribute these events, and to do that for Q fever, the definition was based on the CT value in the qPCR. So if placental tissue, vaginal swab, or fetal sample was positive by IS 1111 in the PCR, but with a CT of less than 27, that was considered a presumptive diagnosis for causation of that abortion event. And then the numbers drop quite a lot, but there are 6 abortion events that were attributed to Q fever in this population. 5 of those were in goats, so that's about 5% of the goat events, and there's also 1 in sheep, none in cattle here. And this is in the context where about 20% of all of the abortion events were attributed to one or other of the pathogens investigated. And just to give a bit of an idea of the frequency of abortion events in these populations. So this table shows the predicted number of abortions per 100 cows by setting. And this again is work by Ward Glanville. So in the pastoral area, we've got a prevalence about 9% per 100 cows. So, and we've got much more frequent abortion events in the pastoral areas as compared to agripastoral and smallholder. This is all for cattle, but we see similar sort of patterns across the systems, and the prevalence of reported abortion events is higher in sheep and goats. So these frequencies are much higher in sheep and goats as well. So these production impacts of livestock abortion and the proportion of those that is attributable to C. burnettii are borne predominantly by the more vulnerable pastoralist populations. Then coming back to the map, locating us again and coming onto the human data that have come through these different platforms. So looking at exposure, disease and risk factors in people. The data that we're using here come from these red dot locations. So that's the back zoo and seeds cross-sectional populations. And these are apparently well members of livestock keeping households that we've sampled. And then we've got these two febrile cohort locations, KCMC, which is a hospital in Moshi serving, it's a referral hospital, but its catchment is quite urban, although it does get cases coming from much wider areas. And Engelin, which has got a pastoral population catchment and quite a local catchment. So samples from all of these different populations have been tested by IFA at CDC, and I'm going to talk about phase 2 antibody results specifically. So it's quite a busy table again. So in these two febrile population studies, by collecting acute and convalescent phase samples, so acute sample collected at the time of presentation, then convalescent some weeks later, we're able to look at defining acute Q fever cases based on a 4-fold rise in IFA titer, and that's a phase 2 titer. And in the Moshi population that was studied 2007 to 2008, there's about an 8% percentage of that population that can be classified as having acute Q fever based on that 4-fold rise. In the Engelin population, where we might have expected a higher proportion of acute Q fever because of all of the evidence that we have that shows more coxiella transmission and exposure in the pastoral populations, we actually just see 4% there. So 6 out of our 147 patients with paired sera meet this criterion of 4-fold rise. I interpret this with a little bit of caution though, because patients present quite late to this hospital in the course of an illness. And so our ability to see that dynamic rise in acute convalescent samples is reduced. And the other thing that we've looked at across these populations and also the cross-sectional population is just a metric of exposure. So this we're defining as an IFA phase 2 titer of 1 in 64 or greater, which I think maps to one in the previous talk. And then we've got about 17% of the total population of febrile people presenting in Moshi meet that criterion for exposure. And a very different story, essentially half of the population and all the other, half of the individuals and all the other populations meet this 1 in 64 exposure criterion. So half of the people presenting to this hospital in Engelin in the pastoral area have been exposed to Q fever, but also about half of the people who come from livestock owning households in both pastoral and non-pastoral areas have been exposed to Q fever by the same criterion. So in these different settings, we've got sort of somewhere between, in different studies over different years, 5–10% of acute fevers in febrile populations that can be attributed to or classified as acute Q fever. Most of that is, as I say, sort of clinically invisible at the moment, unfortunately, based on surveillance and testing opportunities in Tanzania. But that's happening in both urban and rural catchments. We've got really high levels of exposure in all of the populations that are livestock owning. And we're trying to tease apart the risk factors for exposure in those people, and those are boiling down, unsurprisingly, to livestock contacts, handling abortion products, raw dairy products, but so far we're not seeing a clear signal as to which species that's most likely associated with. And shows sort of the difference between these proportions and the relationship of these proportions in this urban population and the pastoral population, we're working as well to try and tease out the difference between risk factors for exposures and risk factors for illness. And that's ongoing, so I can't tell you the answer there. Just finally, just to summarize what we know so far about genotyping of any of this Q fever or Coxiella. So pretty much everything that we've got from these different platforms that's been qPCR positive, we have attempted some typing from. So on the livestock side, and also from some rodent spleen extracts that I haven't mentioned yet, but come from some of the same areas as the cross-sectional back zoo study. And this is work by Kath Allen and Theonest and Bauer. Anything that came through PCR positive from those different populations, we sent lower CT samples to the team in Marseille, so Oleg Melyanikov and colleagues, to apply their multispacer sequence typing approach. Unfortunately, we only got partial typing in most cases. So what we did get, we've got genotype 55 in their scheme in sheep, genotypes 24 and 55 in goats. Couldn't type anything from cattle. But in those two species and also rodents, we've also got indications of lots of new genotypes. So we've got incomplete typing, unfortunately, so far, but what we have is indicating a diversity of Coxiella. And on the human side, we've worked sort of from the Moshi febrile platform led by Matt Rubat and John Crump, and working with Gil Kirsch at the CDC. They're working currently to try and isolate and type from samples from qPCR positive febrile people. And there's one isolation that sounds like it's coming through that will tell us something, we hope, quite detailed genotyping data from a human case. So to bring this all together, we've got really widespread exposure in multiple livestock species and their owners. Livestock shedding DNA detection is really not rare, but we've got, I haven't really gone into the CT, but we see a sort of bimodal distribution, we think, where we've got a sort of high CTs and detection, and then there's a different story, particularly in the abortion events, with much lower CTs and much higher shedding rates. The determinants of the distribution of Coxiella in livestock is about environmental variables, but also species composition and management variables, and these different settings are obviously defined by multiple characteristics that can influence transmission and presence of seed-borne ETI. In terms of human exposure, we're seeing exposure mapping to livestock, and specific contact activities as would be predicted, but a sort of clearer set of, a less clear set of distinctions in terms of what's differentiating the individuals who go on to suffer disease as compared to individuals who are exposed. So we're trying to undertake more work to look at teasing apart the influence of these pathogen and host factors that might play a role there. So the infectious dose, pathogen concentration, and importance of specific high-risk events at which high concentrations of seed-borne ETI shed in terms of leading to clinical illness in people, but also pathogen subtypes and the potential that some of these subtypes are much more pathogenic is something that we're hoping to be able to get more of a handle on soon. And also trying to think a little bit about what methodologies that we're developing for other zoonoses and can be applied for Q, to try and understand the combined impacts on both livestock and human health and the burdens that this pathogen is having for livestock dependent communities in this context. So just wrapping up with sort of plans, ongoing work and future directions. So these risk factor analyses are in the pipeline for livestock seropositivity, livestock qPCR status, and also the human exposure and Q fever risks that I've mentioned in both the urban and pastoralist populations. Ongoing efforts to achieve typing. Increasingly, there are methods that give us more confidence that we'll be able to type direct from field samples, so we're evaluating those. And keen to explore the influence of this parent diversity in Coxiella that we see in this context, so that we can undertake reservoir and source attribution studies. And then try and bring this all together to think about management and prevention in both human and animal populations. On the clinical side, as I said, Coxiella is pretty invisible currently in terms of human management and case detection. And so guideline development there is something that can be informed by some of this work. And as I mentioned, the methodologies to assess the burden that spans animal and human populations can also be used to evaluate and simulate potential interventions and possible control strategies. So that's another avenue that we're continuing to work on. And at that point, I haven't got enough space to thank individuals, I'm afraid. So again, thank you for the opportunity to speak today. And thanks to the teams that contributed to the generation of all this work. Many, many people represented here. And I've listed key partners across Tanzania, New Zealand, the US, the UK, and of course, thanks to funders as well. And thanks to you for listening.
Thank you so much, Jo. Lovely, insightful presentation there. Really important work. Thank you to all the speakers. If I can invite Ben, Rob, Candice, and Jo to turn their cameras on for our Q&A session. So we've had lots of questions coming through, some really interesting questions. Might start with a couple just specifically for Ben on your project. Question about whether any marine mammal sampling was considered. So with that spread into the North Sea.
Yeah, thank you for that question. That's really good. Actually not, because I did not mention that at the beginning. So our QCAPS, or Q fever consortium, was mainly focused on small ruminants, sheep and goats. And yeah, accidentally we came along with that case. And so we decided, okay, to take at least the cattle, the cats, and the dogs into account. And yeah.
Thanks, Ben. And just a question about the assumed source of the infection on that farm. You had said that the cattle were assumed to be the source. What was the reasoning behind cattle rather than the goats? So in which direction the transmission was?
Yeah, so you have to know that, or under German conditions, so I can only talk about the German farming conditions, that we have a really high Coxiella bonnetti prevalence in our dairy cattle population. There are studies saying up to 80% of our farms, our dairy cattle farms, are positive. And our results from the phase-specific physiology and from the genotyping indicates that the source came from the cattle, because firstly, there was, with the phase-specific physiology, we could show that was at least an ongoing or even a past infection there in that farm. And we identified this cattle-associated genotype, C16, in the cattle herd, but also in the goat herd. And within the goat herd, we could show with the phase-specific physiology a dominance of IgG phase 2, which we all know, also it's similar in humans, that this reflects an acute infection. So we have this past ongoing infection in the dairy cattle herd and this acute infection in the dairy goat farm. And again, we found this cattle-associated genotypes from the isolates from the goats. And therefore, we assume that the cattle with the epidemiological background, that it's widely spread in the German cattle population, in combination with our findings that the cattle were the original source of that Q fever outbreak. And we had another farm down in southern Germany. We had similar findings. So for German conditions, we have to consider the cattle as a source for small ruminants. And therefore, I was quite, I think that's really great data from Jo also showed that in Tanzania, this seems to be not the case. So, and therefore, I think it's quite important to do risk assessments for every country and every region, because it can be always quite different.
Thanks for that, Ben. That leads nicely into another question for Jo. So as Ben presented in Germany, the high rainfall was associated with a low risk of Coxiella. But in Tanzania, the high it was high rainfall associated with high risk. Do you have any potential biological explanations for that association? And how could, obviously, Tanzania and Germany, very different settings. Do you have any comments on that?
Yeah, so it's a puzzle we're trying to get our heads around currently. I suspect it's not the case that high rainfall in Tanzania leads to greater exposure to Q fever. I expect that what we have is a challenge with the analysis that because we've got exposure as an outcome, and I see there's another question about how long antibody persists. What we're using is environmental data that's tied to the period before sample collection, but that exposure for that animal could have happened quite a long time prior to sampling. And so we don't have the ability to tie the development of antibodies in real time to the environmental drivers at the moment with this platform. So my suspicion would be that it's not the case that wet environments drive Q fever risk, but maybe more variable patterns of rainfall over time and periods of dry and wet might be patterns that are associated with where there's higher risk of Q fever. So as I say, so teasing apart these relationships is why we're still working on that risk factor analysis to a large extent. But I think it also relates to sort of the I guess, yes, short answer, I'll stop there. But I don't think we're seeing a completely different pattern. I think we're just trying to figure out the best way to disentangle those relationships.
Thanks, Jo. And do you have some comments on why the pastoral livestock had a higher prevalence than the peri-urban?
I think probably because there's sort of a lack of barriers and lots of opportunities for transmission. So in the peri-urban settings, there's a smaller number of individuals. They're less often managed as multi-species groups, and they're less extensively managed. So in the more extreme peri-urban areas, forage is brought to them. They're not moving around themselves. So they're experiencing a sort of different risk environment. And there's probably more vegetation and tree cover in the areas that they're occupying as well. So sort of windborne transmission might be reduced. At the other end of the spectrum in the pastoral areas, it does get very hot and dry and dusty. Animals are managed in much larger mixed species herds, and they explore a much wider area in their grazing. So there's lots of, if Q's in those populations, the transmission will be much easier and happen more readily, I think, in the pastoral systems.
Thanks, Jo. That makes sense. A question for Rob and Candice, but open to comment from everyone. You mentioned differences between Australia and the rest of the world, and that original idea of Q fever in Australia being different to Q fever in Europe. With all the new knowledge in the world, especially from the large outbreak in the Netherlands, do you have any comments about, what your thoughts are now about Australia, Q fever in Australia, and whether it's different from that in Europe?
Yeah, again, it does seem to be different. Again, yeah, the Dutch outbreak was quite different from the French data. In fact, they're almost polar opposites, and Australian data seem to sit in between the two, interestingly, our experience. But we're finding that the serology is the other thing that it doesn't seem to match. And I think also the Dutch have brought that up as well, that they weren't happy with the French serology. So, we may find that it does differ. I think we're trying to work out genetically about the organisms here as well, how much they differ from the ones in Europe as to whether that does anything as well. But yes, we are seeing differences in illness, even up and down Australia. There's pockets where you get severe illness, and then there's other bits where there's very little severe illness, and there's a significant amount of Q fever by seroprevalence studies. So we are seeing that it's not just country to country, it's in smaller regions too that we seem to be some variations. So that's what we're hoping in the next few years to be able to sort of elucidate whether that is true or whether it's just, you know, a statistical artifact that we're seeing this. But yeah, and that's what we're trying to prove. And, you know, we can't really answer much more at the moment till our studies are getting their fruition. Candice, did you have anything to add?
Yeah, no, just I think there was another question in the chat that might be coming up about differences in testing. So obviously we do, yeah, Robert and I see the tip of the iceberg, people that are sick enough to be hospitalised and have those complications. So there, you know, it may be in a little bit to do with testing.
Yeah, we think so, and it varies. There's some GPs in particular are really good at testing and picking up Q, and then there's others that really, you know, don't know much about Q, so they just don't never test.
Thanks, Rob and Candice. A question for Ben that just popped up about whether Germany has an integrated One Health surveillance system that captures Q fever data for humans and animals.
That's a brilliant question, actually. We're working on that. So Q fever is a reportable disease in humans and in animals in Germany. We have two different systems. So one is collecting the data from the animal source and the other one is collecting the data from the human source. And this is one of our aims, to get both systems together. At the moment, we are struggling with the data protection legislation. So, but this is one of our aims to do that. And what we did now was at least to release, or we will soon release, in Q fever guidelines, which consider both parts, both authorities, the human and the veterinary authorities. So they will work closer together in case of a human or of an animal Q fever outbreak.
And Rob and Candice, do you have any comments on sort of blue sky thinking of what's on the horizon for Australia in that regard?
Yeah, unlike Germany, it's not notifiable in animals in Australia, so that makes it very difficult. So we rely on sort of voluntary notifications, and obviously that's not a complete picture, but there are a lot of illnesses which would benefit with this kind of big data and One Health approach to surveillance. So I did a lot of work around Japanese encephalitis virus, and I can echo Ben's comments about the difficulties, even in the midst of you know, a very novel event and significant outbreak, the difficulties in navigating through the legislations, particularly when you've got the different jurisdictions in Australia all having their own unique, you know, nuance to the legislation that you have to work through. But yes, that would be the aspiration, and I think we can get there. It just requires a commitment to work through those barriers, and that's I think the benefit of QFIG, that we have people across the jurisdictions and across the different sectors to be able to add their knowledge and to be able to navigate through that process.
Yes, yeah, we're working on instead of Q fever currently being state-based in terms of whether it's notifiable or not, and having different data fields and data being captured in each state. We're trying to have it all standardized, and in fact to be able to turn it into a national registry.
Joe, do you have any comments on One Health's capture of data for Q fever and coxiellosis, either in the UK context or the Tanzanian?
Sure. So we've actually started doing some work recently in the UK, and the legislation here has changed. So human cases are reportable, but livestock cases have just become notifiable here, related to alignment with European legislations that have been necessitated by recent political changes in the UK. So there is much more sort of proactive sample events happening now, and so pictures of those abortion events essentially prompting reports and collection of materials will lead to more awareness, I think, of what's going on in the livestock populations in the UK. On the Tanzanian side, there's limited surveillance, sadly, in both livestock and human sides currently. That's partly limited by access to diagnostics. So it's quite invisible at the moment. And outside of research studies, there's very little data available.
Thank you very much. And I know there are quite a few other questions, but we are out of time. So thank you very much. And I'll pass back to Jocelyn.
Great. Thank you so much, Rebecca. I might just ask our organising committee to please turn your cameras on just for the close, if that's okay. Thank you so much, Rebecca, for moderating those questions. Just on behalf of the University of Sydney Infectious Diseases Institute, AVA, ASID, Queensland Health, a huge thank you to our speakers for joining us, for sharing your insights. I can see lots of nods from my colleagues. I know that you're dialing in from different time zones, and we truly, truly appreciate your time and expertise. And a huge thank you to our organising committee. You know, this is the joys of working collaboratively across different organisations, and a huge thank you, Ruth, Kate, Catherine, Mike, Rebecca, for pulling this webinar together. Much appreciated, certainly from Sydney ID. And thank you everyone for dialing in. I hope you all enjoy the rest of your either morning or evening, depending on where you're dialing in. And please do stay in contact. Do contact us if you want any further information or any questions for our speakers as well. All right, and on that note, thank you everyone, all the best, and we'll end the webinar now. Thank you.